Toward this end, we will elaborate on the incredible taxonomic and ecological diversity of insect herbivores and their variable feeding styles, investigate the barriers e.g., nutrition and allochemistry that plants pose to herbivore attack, explore the counterploys herbivores have evolved to overcome plant defenses, visit plant defense theory, determine how herbivores and plants affect each other's distribution and abundance, and examine how such information might be exploited to better manage pest herbivores in agricultural and forest systems. The latter is particularly crucial, given that billions of dollars of potential crop yield are lost directly, herbivora, and indirectly, vectors of plant pathogens, to the feeding activities of insect herbivores, Allard et al. 2003, Ork 2006. 4.1 Taxonomic occurrence of phytophagy. At least half of the estimated 2 to 10 million described species of extant insects are herbivores. Phytophages feeding on living plant material, Southwood 1973, Spate et al. 1999, 2008, Gullin and Cranston 2005, Triplehorn and Johnson 2005. Moreover, fossil evidence for the occurrence of phytophagy e.g., herbivora, leaf mines, galls and the Galeriosoph wood borers dates far back in geologic time with numerous records in the Triassic, 220 Maya, and Carboniferous, 330 Maya periods, suggesting that this feeding style is indeed an ancient one. Labandera and Phillips 1996, Labandera 2002. Despite the richness of phytophagous species, the habitat herbivora occurs predominantly in only 9 of the 29 orders of insects: Orthoptera, grasshoppers, androlatives, Phasmatodia, stick insects, Thysanoptera, thrips, Hemiptera, e.g. true bugs, leafhoppers, planthoppers, aphids and scale insects, Socoptera, bark lice, Coleoptera, beetles, Hymenoptera, sawflies, Lepidoptera, butterflies and moths, and Diptera flies. Notably, most species of Lepidopteron Phasmatodia, greater than 95%, and the majority of Orthoptera, Thysanoptera and Hemiptera taxa, greater than 80%, are phytophagous, with a lower incidence of herbivora in the Coleoptera, approximately 35%, Diptera, approximately 30%, and Hymenoptera, approximately 15%. If one includes insect species that consume dead or dying plant material, detritivores, decomposers and shredders, in the category of herbivores, then the prevalence of phytophagy increases substantially. His feeding habit occurs in the 3 orders of non-insect hexapods Protura, Dipleura and Collembola, as well as in 16 orders of insects. Of the terrestrial detritivores, the most noteworthy by far are wreath-wood-feeding Isoptera, termites, whereas in aquatic systems Trichoptera, caddisflies, Plecoptera, stoneflies, and Diptera flies often dominate the feeding assemblage. Of the terrestrial detritivores, 4.2 Diet breadth, feeding strategies and herbivore guilds. Most plant species support complex assemblages of herbivores that collectively exploit almost every plant part, figure 4.1. Synthesizing the incredible diversity of feeding styles and foraging strategies of insect herbivores is a daunting task, but can be simplified by categorizing herbivores according to their diet breadth, host plant range, and feeding guild, a group of species exploiting the same resource in a similar manner. Sensu Root 1973, 2001. Regarding host range, insect herbivores can be monophagous, specialists that feed on a single plant. Lacebug. Leaf galls. Shoot moth larvae. Twig galls, twig girt. Species are plants in the same genus, oligophagous, species that feed on plants in several genera, but within the same family, or polyphagous generalists that exploit plants in more than one family, Strong et al. 1984a, Bernays and Chapman 1994. Using swallowtail butterflies as an example, the Oregon swallowtail Papilio oregonius is monophagous, feeding exclusively on the composite Artemisia dracunculoides throughout its range. The short-tailed swallowtail Papilio brevicata is oligophagous, feeding in several genera of plants in the Apiaceae, and the Anis swallowtail Papilio zelicon is polyphagous, exploiting 69 plants in 32 genera in 2 plant families, Thompson 1998. The fall webworm Hyphantriacunea and gypsy moth Lymantria dispar are extremely polyphagous, feeding on over 600 plant species representing dozens of plant families, Miller and Hansen 1989. Complicating matters, soft diet breadth determination is the fact that herbivores can be locally monophagous, but geographically polyphagous, whereby they specialize on different host plant taxa elsewhere in their geographic range, Singer and Wee 2005. Historically, the perception has been that most insect herbivores are monophagous, greater than 70% of species, but this view is based largely on regional assessments of specific taxa such as aphids, planthoppers, butterflies and agromyzid flies, mostly from temperate latitudes, Wilson et al. 1994, Dixon 1998, Jans et al. 2001. Recent evidence for assemblages of tropical insect herbivora suggests that levels of monophagy may be lower than temperate estimates, at least for some taxa. Bassett et al. 1996, Maudsley and Stork 1997, 1 Degard et al. 2000. Later in this chapter, factors that enhance or constrain diet breadth, influence host shifts and promote speciation and diversification will be considered. Insect herbivores can be characterized by their feeding strategy or guild, and indeed they exhibit an incredible array of feeding styles on living, dying and dead plant resources. Kirby 1992, Gullin and Cranston 2005, figure 4.2. Feeding guild is ultimately affected by a variety of factors, including mouthpart type, chewing versus piercing sucking, the microhabitat where herbivores feed, e.g., leaves, stems, bark, roots, fruits, seeds, dead wood, detritus and fungi, and how plant material is manipulated or processed, e.g., leaf tears, leaf rollers, gall formers, shredders, collectors and scrapers. For convenience, feeding strategies can be grouped into more general categories such as chewers versus sap feeders or free-living feeders, exophages, and concealed feeders, endophages. Notably, it is important to distinguish particular herbivore guilds, because they often respond differently to plant nutrition, allochemistry and natural enemy attack, e.g., Gross 1991, Inba et al., 1999a, Hubbardy and Dino 2004. Of the free-living chewers, those that feed in exposed locations on the plant, e.g., on leaves, flowers, pollen, seed heads and fallen seeds, Lepidoptera and Coleoptera are by far the most diverse and abundant, followed by Orthoptera, grasshoppers, Hymenoptera, sawflies and ants, and Phasmatodea, stick insects, Gullin and Cranston 2005. Many chewing insects also feed in concealed locations within living, dying or dead plant tissues. Important guilds of concealed feeders include leaf tears, Lepidoptera, leaf rollers, Lepidoptera, leaf miners that feed internally between the upper and lower epidermis, Lepidoptera, Coleoptera, Diptera, Hymenoptera, stem borers, Lepidoptera, Coleoptera, Hymenoptera, wood borers, Coleoptera, Lepidoptera, Hymenoptera, that feed within the branches or trunks of woody plants where they consume the bark, cambium, sapwood or heartwood, fruit borers, Diptera, Lepidoptera, and seed pod borers that feed internally within seeds or seed pods, Coleoptera. Lepidoptera, Hymenoptera. By their feeding and oviposition activity, mandibulate herbivores, Hymenoptera, Diptera, Lepidoptera and Coleoptera also induce the formation of galls, structures arising from aberrant plant tissue growth, in which they reside. The diversity of gall sizes and shapes produced by gall inducers is impressive, and galls can be induced on most plant tissues. Mandibulate root feeders, Lepidoptera, Coleoptera. A. E. G. Diptera can be considered concealed feeders simply because they reside in the soil. However, some species feed internally within roots as borers, some Lepidoptera, whereas others feed externally, Coleoptera such as scarab beetles and weevils. Free-living sap feeders, Hemiptera, Thysanoptera feed by inserting their stylets into various plant tissues, and they are categorized accordingly as phloem feeders, e.g., aphids, planthoppers, treehoppers, leafhoppers, scale insects, xylem feeders, cicadas, spittlebugs, and epidermis mesophyll, parenchyma feeders, heteropterans and thrips, that insert their mouthparts into non-vascular tissues, Gullin and Cranston 2005. Numerous sap feeders are also notorious gall inducers, Hemiptera and Thysanoptera, and like their mandibulate counterparts, they, aphids, psyllids and thrips, induce an incredible variety of gall architectures. Several groups of sap feeders, aphids, mealybugs and scale insects, also feed externally on roots beneath the soil surface. In aquatic systems, feeding guilds of herbivorous, detritivorous insects are pigeonholed more into functional groups, Merritt and Cummins 1996, Barber et al. 1999, Gullin and Cranston 2005. There are mandibulate herbivores, Lepidoptera, Coleoptera, Diptera, and a few sap feeders, Hemiptera such as waterboatmen, that feed externally or internally on living macrophytes or algae. Shredders, some Plecoptera, Trichoptera, Coleoptera and Diptera, feed in living or decomposing plant tissues. Collectors feed on plant fragments and decomposing bits off organic matter smaller than those usually consumed by shredders. Collectors are often divided into filter feeders that strain minute particles from the water column, e.g., blackfly larvae and net-building Trichoptera, and gatherers that feed on organic matter on the streamed, several Ephemeroptera, Coleoptera, Trichoptera, Diptera. Notably, shredders break up detritus into smaller fragments, making it available for collectors. Scrapers, Ephemeroptera, Coleoptera, Trichoptera, Lepidoptera and Diptera, graze on surface vegetation or on algae thatis attached to submerged substrates. Many of the feeding guilds of aquatic insects are omnivorous and consume a variety of microorganisms along with the plant material they ingest. Terrestrial detritivores, decomposers and deadwood feeders, e.g., Colembula, Isoptera, Blatodia, Coleoptera, are not often subdivided into feeding guilds, even though they occupy a huge diversity of microhabitats above and below the soil surface, Kirby 1992. Perhaps part of the difficulty in sorting soil and wood-dwelling groups into feeding guilds is that, like their aquatic counterparts, many groups are omnivorous, consuming various combinations of detritus, fungi and dead arthropods. Nonetheless, there are analogs to shredders and gatherers in that larger species, e.g., Isoptera, Coleoptera, process detritus into smaller pieces and fecal material that can then be handled by smaller consumers, e.g., Colembula, Gullin and Cranston 2005. From this discourse, it would be wrong to conclude that herbivorous insects are easily pigeonholed into discrete feeding guilds. For instance, within taxonomic groups there are species that feed in different niches, such as aphids on leaves, stems, bark and roots, Dixon 1998, and Lepidopteran representatives that can be assigned to virtually all mandibulate feeding guilds like free-living folivores, leaf miners and rollers, wood borers, seed feeders, detritivores and even predators, Covell 1984, Powell et al. 1998. Moreover, within a single species, there can be changes in feeding guild throughout development such as occur when miners shift to become free-living folivores and when sap feeders switch from mesophyll to phloem feeding, Powell et al. 1998, Lamp et al. 2004. Nonetheless, categorizing herbivores into specific feeding guilds lends organization to a diverse array of feeding strategies that exists for insect herbivores. During the course of evolutionary time, phylogenetic analysis reveals changes in the feeding strategy of numerous herbivorous insect groups, e.g., from concealed to external feeding in the Lepidoptera, Powell et al. 1998, and later in this chapter we will explore underlying causes and the opportunities such shifts offer for radiation and diversification. 4.3 Plant barriers to herbivore attack. Even though half of the world fauna of insects is phytophagous, the restricted occurrence of phytophagy is a predominant feeding habit to only 9, approximately 30%, of the 29 insect orders. Southwood 1973, Odegaard 2000, Gullin and Cranston 2005 suggests that plants have evolved formidable barriers to insect attack. These barriers include nutritional constraints, mechanical and allochemical defenses, defensive forces of natural enemies, and features of plant phenology and spatial distribution that render plants inherently difficult to exploit. We will explore each of these obstacles in due course, but suffice it to say for now that once plant barriers are overcome, the evolution of phytophagy vastly accelerates. Herbivore diversification, Mitter et al. 1988, 1991, Winkler and Mitter 2007. 4.3.1 Plant nutrition, ecological stoichiometry and constraints on phytophagous insects. Ecological stoichiometry, the study of the relative balance of key elements in organisms from different trophic levels, provides an integrative approach for analyzing plant-herbivore interactions and specifically the constraints that nutrient-deficient food places on consumers. Elser et al. 2000, Fagan et al. 2002, Sterner and Elser 2002. All organisms are composed of these same major elements, namely carbon, C, nitrogen, N, and phosphorus, P, but the relative balance of these elements differs dramatically among organisms occupying different trophic levels. Importantly, nutritional imbalances created by organisms feeding at lower trophic levels on nutrient-deficient N and P food can severely hamper their ability to meet nutrient demands, grow and reproduce. For example, insect herbivores and detritivores have strikingly higher nitrogen, approximately 10% N, and phosphorus, 0.5% P, contents than their host plants, approximately 2% N, 0.05% P, or detrital resources, approximately 2% N, 0.03% P. Elser et al. 2000, Fagan et al. 2002, Cross et al. 2003, Dino and Fagan 2003. Historically, the stoichiometric mismatch in N content % and C, N ratio between plants and insect herbivores has been recognized for decades as imposing fundamental limitations on nitrogen acquisition. McNeil and Southwood 1978, Matson 1980, White 1993, Amak and Leather 2002, Matsumura et al. 2004, Figure 4.3 AC. Similarly, phosphorus limitation has been shown to have widespread effects in aquatic systems, Sterner and Elser 2002, but only recently has it been shown to adversely affect terrestrial insect herbivores, Shade et al. 2003, Perkins et al. 2004. In one case where N and P limitation have been compared in the same insect, the Planthopper Procaliciadalis, N limitation imposes more severe constraints on growth, Hubbard and Dino 2006b, but more studies are needed to confirm any general pattern. Phloem sap. B. The nutritional mismatch between plants and herbivorous insects persists even though there is considerable variation in the nitrogen content of the different plant groups and plant tissues they exploit. Matson 1980, Slansky and Schreiber 1985, Fagan et al. 2002, Figure 4.3a. Of all plant tissues, deadwood and the sap of vascular tissues, xylem and phloem, haveth lowest concentrations of nitrogen, whereas reproductive structures, especially seeds, have the highest reported concentrations. The nitrogen content of detritus is also exceptionally low, but can be enhanced when it is enriched with fungi and bacteria. Slansky and Schreiber 1985, Cross et al. 2003. Overall, phytophagous insects face two obvious problems. Not only must they obtain critical nutrients N and P from nutrient-poor food, but they must also process and eliminate excess amounts of carbon in doing so. Rabin 1983, White 1993, Elser et al. 2000. For active consumers that use carbon-based energy for foraging or dispersal, the need to eliminate carbon to achieve mental balance may be less. Sterner and Elser 2002, Dino and Fagan 2003. Nitrogen and phosphorus limitation have been featured in studies of phytophagous insect nutrition due to their fundamental roles in protein and RNA synthesis, and maintaining elemental balance. C. N. Cheeky smiley face in an organism is essential for metabolism and cell function. Sterner and Elser 2002. Despite elemental mismatches between herbivorous insects and their host plants, herbivores are able to maintain their elemental body composition, at least to some degree, via homeostatic mechanisms. Homeostatic regulation in phytophagous insects occurs by the selective uptake, assimilation, storage and excretion of nutrients. Darchambeau et al. 2003, Trier and Matson 2003. A noteworthy case involves sap feeders, hemipterans such as aphids, leafhoppers and scale insects, that feed in phloem or xylem tissues where they encounter very low concentrations of nitrogen and much higher levels of sugars in the cell sap. Dixon 1998. Homeostasis is achieved by the selective retention of amino nitrogen and elimination of excess sugars as honeydew, a process that is achieved via a unique midgut arrangement known as a filter chamber whereby most sugar-rich liquid bypasses the absorptive midgut and is excreted. However, homeostasis does not occur without cost, which explains why there are such severe growth penalties when herbivores develop on nutrient-poor foods. Sterner and Elser 2002, Raubenheimer and Simpson 2004. Given that herbivorous insects are fundamentally nitrogen limited, it is not surprising that elevated host plant quality %N, either that which occurs naturally or via nitrogen fertilization, can promote increased growth, reproduction and remarkable population outbreaks. McNeil and Southwood 1978, Matson 1980, Cook and Dino 1994, Herms 2002. However, not all feeding guilds respond similarly to elevated plant nutrition, nor do species within the same guild. Schreiber 1984, Kaido Diaresis et al. 1996, Amak and Leather 2002. In general, sap feeders e.g., aphids, planthopers, leafhoppers, scale insects and murid plant bugs show consistent population increases on N-enriched host plants, whereas chewing herbivores e.g., beetles, caterpillars and sawflies range the gamut of responses including increases, decreases and no change. McNeil and Southwood 1978, Matson 1980, Strauss 1987, Kaito Diaresis et al. 1996, Amack and Leather 2002, Dino et al. 2003. Unlike mandibulate herbivores, sap feeders may be more responsive to enhanced plant nitrogen because they feed in vascular tissues, where they benefit from increased soluble nitrogen and yet avoid elevated levels of OFN-based allochemicals, e.g. HCN, that are compartmentalized in other leaf tissues. Raven 1983, Huberty and Dino 2004. Likewise, the high reproductive potential characteristic of many sap feeders may promote their escape from natural enemies on N-enriched host plants, a factor that can compromise potential population increases of slower-growing herbivores. Within the same feeding guild, e.g., leafhopper sandelphacid planthoppers, variable population responses of herbivores to N-enriched host plants have been attributed to species-specific differences in colonization ability, feeding compensation and the level of plant nitrogen at which maximum nitrogen utilization efficiency is achieved. Prestige 1982, Prestige and McNeil 1982, Dino et al. 2002, Huberty and Dino 2. Within Although responses of insect herbivores to plant nitrogen can be compromised at the population level by other factors, there is widespread support for the adverse effects of nitrogen limitation on performance at the individual level. Strong et al. 1984a, White 1993, Amack and Leather 2002. There are many examples showing that phytophagous insects in general survive better, grow faster, molt into larger adults and are more fecund if they develop on nitrogen-rich host plants. Their increased performance on N-rich plants is often attributed to increased feeding rates and enhanced nitrogen assimilation and growth efficiencies. Matson 1980, Slansky and Schreiber 1985, Amack and Leather 2002. Moreover, the extremely slow growth rates, years, of dead wood borers, cerambycid and buprestid beetles, moths and wood wasps, several aquatic detritivores, stoneflies, and some xylem feeders, 17 and 13-year cicadas, all groups that feed on extremely nitrogen-poor food resources, provide further testament to nitrogen limitation. Iverson 1974, Pritchard and Burt, 1987, Motomori et al. 2001. Although ecological stoichiometry provides a broad context for highlighting the general nutritional constraints that consumers face, it does not emphasize important details such as the appropriate form of the macronutrient ingested, e.g., nitrogen-containing toxins, amino acid balance in the diet, or the need for non-synthesizable nutrients, e.g., sterols required for molting hormone, and limiting trace elements and water-soluble vitamins, e.g., thiamine, riboflavin and ascorbic acid, all of which are necessary for metabolism and development. Prestige and McNeil. 1982, Bernays and Simpson 1990, Singer et al. 2002, Singer and Bernays 2003. Moreover, the water content of plants affects cell turgor pressure and the ability of sap feeders to access plant nitrogen. Huberty and Dino 2004. Similarly, the nitrogen utilization efficiency of chewing herbivores is often diminished under conditions of lowered foliar water content. Slansky and Schreiber 1985. Thus, plant conditions such as water content and other dietary factors can hinder herbivore performance beyond the general constraints imposed by contrasts in macronutrient stoichiometry with their host plant. Schreiber and Slansky 1981. 4.3.2 Coping with nutrient-deficient host plants. Because insect herbivores are inherently nutrient-limited, they have evolved a variety of adaptations or feeding strategies that maximize encounters with nutrient-rich resources or buffer them against nutrient deficiencies in their diet. McNeil and Southwood 1978, Cook and Dino 1994, Carbon and Agrawal 2002. These adaptations can be organized into 6 general categories. 1. Feeding compensation. 2. Selection of nitrogen-rich feeding sites and/or diet mixing. 3. Life cycle synchronization with nutrient-rich resources. 4. Manipulation of plant physiology by forming nutrient sinks. 5. Obtaining nitrogen from non-plant sources. 6. An evolutionary shift in body nutrient composition. Feeding compensation. By increasing their feeding rate on nitrogen-deficient plant resources, insect herbivores can partially offset the problem of satisfying their nutrient demands, a phenomenon which occurs in a wide diversity of sap feeders, hemiptera, aphids, planthoppers and leafhoppers, chewing herbivores, Orthoptera, Coleoptera, Lepidoptera and Hymenoptera, and detritivores, Plecoptera and Trichoptera. Iverson 1974, McNeil and Southwood 1978, Bernays and Simpson 1990, Simpson and Simpson 1990. Slansky 1993, Young and Jorn 1994, Kaus et al. 1999, Swan and Palmer 2006. Feeding compensation, however, is not a completely effective solution to problems of nutrient acquisition because of physiological constraints such as gut capacity and throughput time that limit the degree to which eating more can compensate for eating nutrient-poor food. Simpson and Simpson 1990. For instance, correlated with increased feeding rate in nutrient-deficient food are a shorter gut retention time and often decreased digestion and nitrogen assimilation. Moreover, increasing consumption rate to compensate for a deficiency in a nutrient may lead to an excess of other carbon-rich compounds or dietary toxins that can negatively affect growth and survival. Slansky and Wheeler 1992, Amack and Leather 2002. Also, if feeding compensation results in delayed development, then herbivores may also experience higher exposure to natural enemies, the so-called slow growth high mortality hypothesis, Price et al. 1980, Benry and Dino 1997, Kaplan et al. 2007. Selection of nitrogen-rich feeding sites and diet mixing. Specialized sap feeders that are generally less affected by compartmentalized allochemicals show very strong preferences for nitrogen-rich feeding sites, Matson 1980, Cook and Dino 1994, Amack and Leather 2002. Feeding sites where high concentrations of nitrogen occur include actively growing meristems, young leaves, inflorescences, pods, seeds and senescing leaves, and sap feeders such as aphids often aggregate at these sites, where their performance is dramatically enhanced. Many sap feeders also shift their feeding position from low to high nitrogen sites with the seasonal decline in leaf nitrogen that occurs in many plant species, McNeil and Southwood 1978, Schreiber and Slansky 1981. A frequent spatial shift is from leaves to more nitrogen-rich inflorescences or seed heads with the onset of flowering. Chewing folivores, Lepidoptera, Coleoptera, Hymenoptera, show more variable responses than sap feeders with regard to selecting the most nitrogen-rich feeding sites on a plant, namely young compared to mature leaves, Raup and Dino 1983. In general, specialist defoliators, which are better adapted than generalists to deal with allochemicals concentrated in young leaves, show stronger feeding preferences for nitrogen-rich young leaves, but there are exceptions, Raup and Dino 1983, Amack and Leather 2002. Although performance is often potentially greater on nitrogen-rich plant tissues, specialized herbivores do not always select such sites for oviposition or feeding because performance is compromised by high concentrations of performance-reducing allochemicals, increased risk of enemy attack, or because herbivores forage selectively elsewhere on the plant for toxins that they sequester for defense, Damon 1987, Amack and Leather 2002. Among polyphagous herbivores, such as many grasshoppers and lepidopterans, there is certainly evidence that high nitrogen diets are selected to meet nutrient demands, Bernays and Simpson 1990, Jorn 2000, Amack and Leather 2002. Similarly, many detritivores like the leaf-shredding caddisfly Hydatophylax selectively colonize high nitrogen litter over less nutritious choices, Cummins and Klug 1979, Motomori et al. 2001. However, diet mixing, feeding in more than one plant species or food resource, is a common feeding strategy in many herbivorous insects, e.g., numerous grasshoppers and caterpillars, and detritivores as well, e.g., stoneflies and caddisflies, Jorn 2000, Bemer et al. 2001, Singer and Bernays 2003, Swan and Palmer 2006. Some herbivores, e.g., many grasshoppers, and detritivores, e.g., stoneflies, and predators for that matter, in fact grow faster and exhibit higher fitness on mix compared to single-resource diets, McFarlane and Thorsteinson 1980, Mainz et al. 2005, Swan and Palmer 2006, whereas others do not, several heteropterans and lepidopterans, Bernays and Minkenberg 1997. Diet mixing theory, in partial contrast to ecological stoichiometry, emphasizes achieving dietary balance by foraging selectively on limiting macronutrients such as protein and carbohydrate, Bemer et al. 2001, Raubenheimer and Simpson 2004, Raubenheimer and Jones 2006. Grasshopper species, for instance, grow best on diets composed of a specific proportion of protein and carbohydrate, their so-called nutritional intake target. When fed diets deviating in protein-carbohydrate composition from their intake target, performance is reduced. So, it is not surprising that grasshoppers forage selectively on a dietary smorgasbord of protein and carbohydrate choices to achieve their intake target, although targets do differ among species. Diet mixing theory extends from optimal diet theory, which itself stems from optimal foraging theory, see in Christensen 2001. The essential predictions of optimal diet theory are that foragers should conform as follows. 1. They should prefer food items that yield more energy per unit handling time. 2. Individuals should drop low-value items from their diet as higher-value options become available. 3. They should obey a quantitative threshold rule as to when specific food types should be included or excluded from the diet, Charnov 1976, see in Christensen 2001. The observation that diet mixing can be more beneficial to consumers than simply feeding on the most nitrogen-rich resource challenges the more singular focus of ecological stoichiometry and has led to a multitude of multifaceted hypotheses to explain diet choice and mixing by polyphagous herbivores and omnivores, Bernays and Bright 1993, Jorn and Bemer 1997, Jorn 2000, Bemer et al. 2002, Singer and Bernays 2003, Raubenheimer and Simpson 2004, Raubenheimer and Jones 2006. Besides achieving nutrient balance, alternative hypotheses for diet mixing include feeding on nutrient-deficient resources when superior alternatives are rare, diluting toxins that are ingested from nutritious food resources, diet sampling to assess optimal resources, and minimizing exposure to natural enemies. Future development of food selection theory should seek to combine the constructs of stoichiometry with the complementary views of diet mixing theory, see Raubenheimer and Simpson 2004. Overall, however, there is overwhelming evidence that nitrogen and phosphorus limitation are pivotal factors directing the feeding strategies of insect herbivores. Life cycle synchronization with nutrient-rich resources in time, diapause, and space dispersal. There is tremendous spatial and phenological variation in plant quality, nutrition, and allochemistry that occurs within and among plant species. McNeil and Southwood 1978, Hunter et al. 1997, Dixon 1998, Amack and Leather 2002. In general, the nitrogen content of plants such as grasses, forbs, and trees follows a distinct seasonal pattern. Leaf nitrogen is highest in spring following bud break, declines rapidly thereafter to a summer low and then rises again during autumn when nutrients from senescing foliage are being translocated to roots. However, among individuals of Thesame plant species, there is remarkable temporal and spatial variation in the onset of this progression on the maximum nutrient content that ultimately occurs. Also, within the same habitat, various plant species peak in nitrogen content at different times. Failure to synchronize reproduction and development with windows of high nitrogen opportunity can have drastic consequences for herbivore performance and survival. McNeil and Southwood 1978, Cook and Dino 1994, Hunter et al. 1997, Dixon 1998. 2 life history traits, namely diaposy and dispersal, allow herbivores to synchronize reproduction and development with optimal plant nutrition in time and space respectively. Dino 1994a. Several of the best examples of nitrogen tracking occur in the sap feeding guild. McNeil and Southwood 1978, Dixon 1998. For instance, population size of the green spruce aphid Eletobium abitinum mirrors changes in the amino nitrogen content of its host, with peak reproduction occurring in spring when trees are most nutritious. Figure 4.4. A second population rise often occurs during autumn and is associated with the mobilization of amino nitrogen and its back translocation to roots. Notably, the production of winged adults Alatae, that can disperse to more nutritious trees elsewhere occurs in summer when there is a precipitous drop in the amino nitrogen content of the resident tree. Other monophagous herbivores also synchronize their life histories with high plant nitrogen by dispersing to more nutritious plants. For example, the salt marsh planthopper Procollecia marginata meets its high nitrogen demands by dispersal, which allows for the escape of deteriorating plant patches and the colonization of nutrient-rich plants in other habitats where offspring performance is enhanced. Cook and Dino 1994, Dino and Peterson 2000. By contrast, its sympatric congener P. dallas is relatively immobile and copes with declining plant nitrogen by compensatory feeding. Hubbardie and Dino 2006a. A morphological trade-off in investment between flight muscles P. marginata and the muscles governing ingestion capacity P. dallas imposes constraints on how these sap feeders cope with declining plant nitrogen. Figure 4.5. Thus, dispersal and feeding compensation may be 2 competing mechanisms for satisfying nitrogen demands that can't both be maximized in the same species. Dispersal allows for avoidance of low nitrogen conditions, whereas feeding compensation permits tolerance until plant nutrition improves. Because the nitrogen content of different plant species peaks at different times of the year, polyphagous herbivores can meet nitrogen demands by dispersing to more nutritious plant species as local conditions deteriorate. McNeil and Southwood 1978, Prestige 1982, Amack and Leather 2002. For instance, the grass bug Leptoptera nodulabrata and the planthopper Jabecilla pellucida switch host grasses from one generation to the next as they track spatial changes in high nitrogen availability. Moreover, host alternation in aphids has been linked to nitrogen tracking. Dixon 1998. Host alternating aphids hatch from overwintering eggs on their primary host, usually a woody tree or shrub. As the amino nitrogen content of their primary host declines, migratory females are produced that disperse to more nutrient-rich secondary hosts, herbaceous plants, where they undergo several asexual generations until plant quality declines. Subsequently, migratory forms are produced that return to the primary host, where they reproduce and give rise to the egg-laying sexuals. The decrease in quality of the secondary host coincides with the autumn increase in leaf nitrogen content of the primary host. Other aphid species such as the sycamore aphid Drepanocyphum platanoides show similar patterns of precise nitrogen tracking, but do so by entering a reproductive diapause in summer and thus avoiding periods of low nitrogen availability. Dixon 1998. Some herbivorous insects that overwinter as diaposing eggs on their host trees, e.g., treehoppers, detect plant cues in spring and synchronize egg hatch with the onset of sap flow, thereby ensuring development on nitrogen-rich leaves. Wood et al. 1990. Other herbivores such as foliar-feeding caterpillars are less able to predict bud break and thus peak leaf nitrogen. Hunter et al. 1997. If larvae hatch just following bud break, they experience high-performance onotrogen-rich leaves. However, if they hatch too early, bud scales preclude access to developing leaves, and if they hatch too late, they incur the adverse effects of declining leaf nitrogen and increasing defensive chemicals, both of which result in lower survival. Manipulation of plant physiology by forming nutrient sinks. Several species of free-living aphids, gall-inducing insects and lepidopterans modify plant nutritional physiology to their own advantage by creating nutrient sinks. Way and Camel 1970, Larson and Witham 1991, Inbaugh et al. 1995, Rahman et al. 2006. When phloem tapping herbivores feed, they altered source sink dynamics of phloem transport by diverting assimilates from neighboring leaves and drawing them toward feeding sites where their performance is dramatically enhanced. Moreover, by feeding in aggregations, some aphids Brevicorina brassicae further facilitate the local accumulation of nutrients and increase the strength of the nutrient sink. Similarly, the developing Galoth, the Lepidopteran Epibulma strenuana, intercepts the normal flow of nutrients and acts as a nutrient sink. Caption for Figure 4.5. Kant. Seed-feeding insects can also manipulate seed development to their own advantage. Von Aderkes et al. 2005. For example, the seed chalcid Megastigma spermotrophus Hymenoptera Torimidi deposits eggs in the ovules of Douglas fir, Pseudosugum and ZC cones. Oviposition prevents the expected degeneration of unfertilized ovules and at the same time induces the accumulation of energy reserves, which larvae require for development.